Hello, welcome to the video on variation in chromosome number and structure. This is designed to complement the lecture material. So I hope you've viewed the lecture and I hope you've made an attempt to work the problem set prior to watching this video. This video will work best if you have attempted the problems first and then use the video to check your work and to get assistance with trouble spots. So let's start off with some vocabulary. When we think about variation in chromosome number, we often think about aneuploidy and polyploidy. Aneuploidy occurs when the chromosome number in an interphase cell is not an exact multiple of a haploid number. For example, in people, we are described as 2n equals 46. So an individual that had only 45 chromosomes, like Turner syndrome, or 47 chromosomes, like Klinefelter syndrome, would be considered to be an aneuploid. We also look at different numbers of sets of chromosomes. And we don't see this in people that are live people. We do see this in spontaneously miscarried fetuses, where we have extra copies of entire sets of chromosomes, like 3 of every chromosome would be triploid, 4 of every chromosome tetraploid, and so forth. We do see this quite often in plants. And in plants, when we see the higher ploidy levels, we have the advantage of extra vegetative growth. So we get these lush vegetative green plants with big leaves and big vegetative parts, or maybe we get very full flowers like the carnations that look like pom-poms. So let's look at a little bit of vocabulary. Corn is a diploid plant. It's described as 2n equals 20. There are not any sex chromosomes in corn. And we're going to complete the table below with the number of chromosomes present in a cell from a corn plant with the following description. And we are assuming all of these are interphase cells. So normally, corn has 20 chromosomes. A monosomic corn plant would be missing a chromosome. So it would only have 19 chromosomes. A trisomic would have 1 extra chromosome. So instead of 20, it would have 21. A haploid corn plant only has 1 of every chromosome type instead of the normal 2. So typically we have a diploid with 20 chromosomes. In fact, that's the next one down here. So a haploid has 10 chromosomes, and it has 1 of each chromosome type. A double monosomic means that the corn plant is missing 1 chromosome from 2 different pair numbers. So instead of having 20 chromosomes, it's missing 2 chromosomes that are different pair numbers. So it has 18 chromosomes. A nullosomic corn plant also has 18 chromosomes, but the 2 that are missing here are both the same pair number. So that's the difference between the double monosomic, where the 2 missing chromosomes are different pair numbers, and the nullosomic, where we don't have any of a particular pair number because both of the missing chromosomes are the same pair number. Now let's move on a bit to chromosome aberrations. In lecture, we talked about 4 main types of aberrations: duplications, deletions, inversions, and translocations. And we talked about several types of each of those. So let's look at this problem. And it says an individual has the 4 chromosomes below. Well, no, I think in this case it has the 2 chromosomes below because I see 2 chromosomes here. So consider that a typo. The periods represent the centromeres and the letters the genes. What type of chromosome aberration is present? So I usually like to start looking at where the differences are between these. So WT, there's a difference right there. And then if I go from the other end, ADG, and here's a difference, either S or K. Now look at this part that's changed here. If I read the one on the left, left to right, and the one on the right, right to left, they're the same. SR centromere K. So this is an inversion. And notice the centromere right here is part of the inverted sequence, and that is a pericentric inversion. It's an inversion because a segment of the chromosome is basically reversed. It's in opposite order. And it's pericentric with an I because the centromere is part of the inverted area. So let's see what would happen if we tried to synapse chromosomes from this individual. Inversions form what we call an inversion loop. And the general structure for the inversion loop is always the same. Synapsis occurs when there are two chromatids attached to every centromere. So this is the second chromatid for this first chromosome. One of the homologous chromosomes makes a loop. The second one goes in, jumps over, and goes out. And here's the second chromatid for that chromosome. Now, having these little lines here indicating where the inverted sequence is really helpful if you want to try to draw this out, because the part that's inverted is on the loop when we label the genes on these chromosomes. The part that is before the inversion is before the loop, and the part after the inversion is after the loop. So I've got WT. And remember, it doesn't matter if you label this one as the one across the top or that one as the one across the top, because when these chromosomes synapse, these genes will line up across from the same gene. So no matter how you do it, it works out just fine. So WT, and then on the loop, I've got SRPM centromere K. So SR. And after the loop, GDA. Now, across from each of these genes, we still have to have the second chromosome labeled, and it will have the same alleles, because remember, synapsis is occurring as a locus by locus pairing up. There we go. And if you notice the way I did it, I put this one as the one reading across the top, WTSRPM centromere K GDA. And this one is the one reading in the middle. So WTK centromere MPRS GDA. Now our problem says, assume we have a crossover between the R and P genes. That's going to be right in here. Remember, a crossover is between non-sister chromatids of homologous chromosomes. So we want to make sure we cross one chromatid that's attached to this centromere and one attached to that one. The easiest way to do that is to do the two middle ones. And we want to figure out what the gametes are. Let's see if I can write it. There we go. Maybe enough room. No, that's a bit hard to read. That one is a P. Okay. I like to start with an N and mark my N's as I do them. And I also use some sort of a marker or color just so I can keep track of what I'm doing. So my first one, WT SRPM centromere K GDA. And it's going to use the top lines. So let me write that down. WT SR. Okay. This is considered to be a viable gamete. It has exactly one copy of each of the genes. Well, let's take the next line down. And I'm going to start with the second one right here. WT SR. Now this is a crossover, cross down, PM, centromere K. And look, it's winding us all the way back down here. Oops, TW. So, let's see. WT SR cross down P, cross down P, M centromere K, TW. And this is not viable. And we know it's not viable because it's missing GDA and it has two copies of W and T. And this one was viable. Oh, let's see. I need a third color. Make orange. And I'm going to start with this third line here. WT K centromere MPRS GDA. So WT K centromere M PRS GDA. This is a viable gamete. So both of these two are viable because it has exactly one copy of each of the genes. And if we think about looking at these, they are the normal order and the inverted order. And actually, we don't know which is the normal order and the inverted order. Now our last one must start at the right because we've used all of our starting points over here. And ideally, it will finish up down there. So let's try the pink line for this one. ADG K centromere MP cross down RS, and it does go all the way back here, GDA. So we managed to use up all of our ends. Now let's see if I can write this down right. Whoops, not with that color. ADG K centromere M PRS GDA. This is also not a viable gamete. And we know that because it has two copies of the GDA and is missing the WT. I encourage you to practice these. There are a few practice problems, and then I believe you have a homework that's kind of similar to this as well. Let's try another chromosome aberration. Now, this individual does have 4 chromosomes, 1, 2, 3, 4. And again, the period represents the centromere and the letters represent the genes. Now you don't know what is normal and what is messed up. So we can just look and see where the differences are. Look around the centromeres. One centromere is G centromere H. There's the other G centromere H. I'm just going to highlight that centromere in pink because those are homologous centromeres. And let's see what's attached to each of these. One has next to the G, F, okay, got that. Oh, and then it's got BC on this one and XC on that one. And going the other way, HLM is the same. Well, let's look at this other centromere. It's between R and S, and it goes S centromere RQ there. But on this side, next to the T, there's a CB, and next to the T, there's an XC. So what happened here is that both of these chromosomes, these are non-homologous chromosomes, these two are homologs, those two are homologs. Each of them broke a little piece off and then the pieces switched. And it doesn't matter which was normal and which was defective to start with. This would be called a reciprocal translocation. So let's just assume that the ones with the pink centromeres are the normal ones, just so that we can, only one with the pink centromere is normal. We'll assume this one and this one are normal. And that gives us one copy of every gene. Then that means these two would be normal and these two, the BC, switch places with the XC. And that's our reciprocal movement between the non-homologous chromosomes. Reciprocal translocations synapse as a cross structure. So we can draw that general cross structure out. And again, synapsis occurs when there are two chromatids on every centromere. So I'm drawing the second chromatid now. And it doesn't matter which of these I begin labeling. I'm just going to start with this one. Where I've made the line, that's like the corner. So BC, and then there's the break point, FG centromere HLM. So I can go B, C, break, F, G, centromere, H, L, M. Almost ran out of room. So let's see, F, G, H, L, M. Now this centromere is the one that I coded as the pink centromere, so I'm just going to make that pink. Now the other one, let's see, my next chromosome, since I've already labeled this part right here, because I know homologous areas have to pair, that would be this one that also has the MLH, that is an H, centromere GF, but then down here it would have XZ. Across from the BC here, I must have the other BC. And then it would go after the break point, so after the corner, TS centromere RQ. Now this one should be easy because it should be exactly what's left. So, and it should be exactly what's across. So Z, X, T, S centromere R, Q. Okay. Now, how are the gametes formed for this? When we have the gametes formed, we expect one of each of these centromeres, so one pink centromere and one non-pink centromere, to go to each pole. And we can think about the 4 ways this can occur. So one way, these two go to one pole. A second way, that means these two go to the pole. Then the opposite ones, the top upper left and lower right go to one pole, and then the last one would be the upper right and lower left. So let's just draw these out. The other part of this question says, which are viable? In order to be viable, we have to have exactly one copy of each of the genes at the end of the day. So I'm going to start by looking at the ones on the left. B, C, F, G, centromere, HLM. And this is our pink centromere. And then the other is B, C, T, S, blue centromere, or S. That's not viable because we have two copies of BC and then we're missing X and Z. So let's look at the other side. If we had gone and taken the right side, MLH centromere, GF, X, C, and that centromere is the pink centromere. And then on this side, we would have had Q, R, blue centromere, S, T, X, Z. Also not viable because now we have two copies of XZ and we're missing the BC. So these two. The other two will be viable. So these two here will be viable. And one way we can remember that is when we take the diagonal chromosomes, we end up with viable gametes. So let's take this diagonal upper left, lower right. B, C, F, G, pink centromere, H, L, M. And then down on the bottom, Q, R, S, T. Right, so that one's viable. Now we have to do the other diagonal, upper right and lower left. So MLH, pink centromere, GF, XZ, and then this one over here, BC, TS, blue centromere, or Q. Okay, so this one are these two right here. Whoops. That one, these two. And this one is these two. Okay. Any questions? Please do feel free to email me if you do have questions. These can be kind of tricky, and it's important that you get used to tracing these lines so that you can answer questions regarding what the pairing would look like and what types of gametes would be formed and which gametes would be viable or not viable. One of the last things we looked at in our lecture was creating interspecific hybrids. And we said that a lot of our crop species have actually come out through that amphidiploid pathway where two lines crossed, they formed a sterile F1, the chromosomes automatically doubled because this just happened naturally in the evolution of some of our crop species. And then we ended up with a fertile amphidiploid that behaved like a diploid. So here we've got two species. Species A is 2n equal 40. Species B is 2n equals 18. They are not related to each other. And that's important because by saying they are not related to each other, it means if we cross them together, the hybrid should be sterile. We should not get any of those chromosomes pairing up. You cross species A and B together to get hybrid X. You then treated hybrid X with colchicine to produce hybrid Y. Then you mated hybrid Y to a plant of species B to get hybrid Z, and now you have to fill out the number of chromosomes present in a somatic cell in interphase in each of the hybrids, the number of univalents present in synapsis of meiosis, and the number of bivalents present after synapsis and meiosis. So let's start by diagramming what happened here. So species A two n equals 40, species B each of these produce a gamete, and let's see, this first hybrid is hybrid X. So half of the chromosomes in species A would be in the gamete, that's 20. Half of the chromosomes in species B would be in the gamete, half of 18 is 9. So hybrid X has 29 chromosomes. And we expect hybrid X to have those 29 chromosomes all as univalents, because remember, our two species are not related to each other, so no bivalents present. Then we added colchicine to get hybrid Y. So 29 times 2 is 58, because colchicine doubles the chromosome number. Now, every chromosome has a buddy, so we don't expect there to be any univalents. A bivalent is 2 chromosomes, so when synapsis occurs, the 2 chromosomes that are partners partner up in synapse. So instead of 58 individual chromosomes, we have 29 bivalents, because each of these bivalents equals 2 chromosomes. Now the tricky part. We mated hybrid Y to species B. Yep. To get hybrid Z. Okay, species B has 9 chromosomes. Hybrid Y has 58. Well, species B has 18 chromosomes, but the gamete has 9. Hybrid Y has 58 chromosomes, so the gamete has 29. So 29 plus 9, that's 38 chromosomes. Put that in our chart. Now, some of the chromosomes, though, in hybrid Y are related to those in species B, because species B is one of the progenitor species of hybrid Y, or ancestral or progenitor species. And it happens to be 9 of them. So those 9 will synapse with 9 of our 29. And of those 38 chromosomes, that means we only had 20 over here that are left to be univalents. Remember, each of these counts as 2 chromosomes, so we have to have a total of 38 chromosomes. 9 times 2 is 18, plus 20 is 38. Okay, I encourage you to walk through this. I think there are some other examples in the lecture and also in your homework that will help you do something similar. The last thing I wanted to point out today is a chart that is already answered for you. And this is designed to help you identify some of the differences between the types of chromosome aberrations. Remember, we have deletions, duplications, inversions, and translocations. And then also, how did they originate? What kind of effect could we have on that particular individual? And then what kind of effect could we have on the offspring? So I encourage you to go through this and make sure you feel comfortable with the qualitative material in this chart. Thank you for your attention to this problem session today. I hope you have a wonderful day, and please contact me if you have any questions at all.